First of all, I would like to give my appreciation. Yeah, I got it. To give my appreciation to the NSG team for hosting this seminar, and it's a great honor to have this opportunity standing here to talk about our story about the CD3 VHH antibody and the development of the T-cell engager bispecific antibody. First of all, let's do a really quick overview of the competitive landscape of the CD3-based T-cell engager. The T-cell engager market represents a really fast-growing segment of the bispecific antibody therapeutics. There are so far 13 marketed bispecific antibody, and 9 of them are the T-cell engagers. In addition to that, there are over 1,000 CD3 bispecific antibody pipelines, over 100 of them are in the clinical and late stage. So here is a summary table for the 8 T-cell, the T-cell CD3-based T-cell engager drug products that are still available on the market. Judging by the table, you can see that the monovalent format, the 1+1, is still the mainstream choice for most of the CD3-based T-cell engager. And in addition to that, the choice of the CD3 arms tend to be medium to low affinities, while the affinity for the TA arms is more diversified. And again, the SP34 is a popular choice for the CD3 arms. All of these 3 features derive from the concerns of the common safety issues of the CD3-based T-cell engager. We will get into more details regarding these particular concerns in the next couple of slides. So when designing a bispecific antibodies, usually 4 aspects need to be considered in advance. The first is the affinity and the valency. We have already briefly talked about in the previous page regarding the CD3-based T-cell engagers. Usually it comes from the consideration of the safeties and the balance between safety and efficacies. In addition to that, the length, the flexibility, the amino acid composition of the linkers is another factors need to be considered for the stability of the molecules and the immunogenicity of the candidates. The third aspect is the target distribution. To be more specific, it's the epitope. There is a theory that in the T-cell engager that the epitope on the TA arms needs to be proximal to the cell membranes to form a immune synapses to facilitate the effective killing of the T cell to the target tumor cells. And the last aspect is the molecular weight. Of course, the larger the molecule is, it has a better, longer half-life, while the smaller it is, it has more tissue penetrations. It's another issue of the balance between the size and the tissue penetration. So, given the complicated design of most of the bispecific antibodies, it also brings several challenges. The first of all is the immunogenicity. This over-engineered structures often leads to the safety risk, and this kind of a safety risk cannot be fully assessed or predicted during the preclinical stage. And the second common issue. Sorry, it's out of battery. Let's try that one.
Yeah, more close.
All right. The second common issue is the developabilities. The formation of the homodimers, the prone to aggregations, and the loss solubilities are the most common things we have seen with these over-engineered molecules. And the last common issue is the manufacture problem. These overcomplicated structures always brings issues like multiple plasmid during transfections, low expression levels, the complicated purification process and the post-production process. All right, next let's do a quick overview of the workflow for a typical discovery process with a T-cell engager, starting with the discovery of the monoclonal antibody for the TAA arms and the CD3 arms. At BsAb, we have multiple options for the antibody discoveries, including single B-cell approach, hybridoma approach, and the phage library approach. This is a typical workflow for the single B-cell based approach for the monoclonal antibody discoveries. We utilize the beacon from previously known as a Berkeley Light. Now it's acquired by Bruker. In this platforms, basically we can facilitate the B-cell screening within 1 day on a microchip at the high throughput fashions. This screening typically includes protein bindings, cell bindings, and the blocking functional assays. After the on-chip screening, the positive B cells can be exported for sequencing and recombinant production for larger scale production and profilings, including the immunization. The whole process from antigen preparation to identification of the functional leads usually takes about 3 to 4 months. The second approach is hybridoma. This is probably one of the most established approach for the antibody discoveries. However, at ProBio, we utilize a slightly different workflow. Instead of doing the subclone, spending like 2 or 3 months doing the subcloning, we plate the hybridoma cells as a monoclonal in the 384 well plates, followed by ELISA, flow cytometry screening, and the cell-based high throughput functional assays. So with this process, we can eliminate the timeline for the subcloning, making the overall timeline from antigen preparation to recover of functional sequence in 3 to 4 months, pretty close to the previous single B-cell based approach. And the last approach is the video display-based library technologies. I believe most of you guys are pretty familiar with it, so I will skip most of the information here. We only have like 20 minutes. So after the identification of your monoclonal antibody leads, the next step we will move on to the engineering, typically including the humanization and affinity maturation. Humanization usually can be done within 3 weeks, including the sequence design, the recombinant production, and the affinity characterization, the kinetics measurements by BioCore, SPR, or BLIs. Regarding the affinity maturation, it really depends on the affinities of your existing candidates and the requirements of your target molecules. At ProBio, we can achieve the improvement of affinity at least tenfold in about 16 weeks. Once the sequence engineering part is done, next we'll move on to the construction of the bispecific antibodies. So in this table, we listed 5 of those most common formats for the T-cell engager bispecific antibodies. From the first to the third, the BiTE, the cross-map, and the DuoBodies are the typical monoclonal, the monovalent 1+1 versions, which are pretty popular in the clinical and the preclinical pipelines. The fourth one, the IgG fused with VHH or single-chain Fv, forms a tetravalent bispecific antibodies, which is not very popular in the T-cell engager field because of the common safety issues. Because if you include 2 copies of the CD3 arms in your bispecific antibodies, usually it will lead to overtoxicities. But we will demonstrate a case studies in the second part to show that it might not be an issue with a proper selection of a VHH CD3 arms. The last one is called the asymmetrical 2+1 format, which is also very popular in recent years for the T-cell engager. Basically, in this format, it utilizes 1 copy of the CD3 arms, the 2 copy of the TAA arms to achieve differential binding of the bispecific antibodies on the cancer tissues over the healthy tissues. After the constructing of the bispecific antibodies, the next step will move on to the cell-based functional validations. Typically, 3 kinds of assays will be considered and included at this stage. The report gene assays, the TDCC assays, and the cytokine release assays. These assays will give you a preliminary evaluation of the efficacies and the safeties of your candidate sequence. When you do the TDCC assays, you have 3 different options for the effector cells. Most of the people commonly use the PBMC, the one in the middle, as the effector cells. But depending on the source of the PBMC, you may experience some fluctuations or noise issues when you're doing the TDCC assays. So that's why people may do some T cell sortings to select out the CD8 positive T cells or the pan T cells as the effector cell for the TDCC assays. So while we are doing the in vitro assays, the developability is another crucial factors needs to be considered in parallel. Typically at ProBio, we will consider doing the developability assessment at 3 different stages. The first stage is when you do the humanization or affinity maturation, the engineering timelines. During this time point, we will consider do the evaluation of immunogenicities, the post-translational modification analysis, and the aggregation analysis. So we can do the engineering regarding these 3 aspects along with humanization and affinity maturation. The second stage is when you do the cell-based assays. In addition to the function assays, you can also do this non-specific binding by BUP ELISA, DNA ELISA, and the self-interaction by ACCs, and also the plasma stabilities. And the third stage is when you have completed all the engineering and the in vitro evaluations. When you start to do the in vivo animal studies, you will generate a large amount of samples to assess the manufacturabilities. Basically, the purpose of this step is to assess the integrity and the stabilities of your molecules under stressful conditions like high temperatures, repeated first salt cycles, and acidic or basic conditions. While we're doing the developability analysis, the animal-based disease models is another crucial steps to give a full evaluation of efficacy and the safeties for the candidate sequence. For the efficacy studies, typically we can apply the syngenic models or the CDX models for T-cell engager. These are pretty common models for the evaluation of efficacies for T-cell engager bispecific antibodies. At the same time, we can do the PK and bioanalysis by either plasma stabilities or in vivo stabilities in rodents, or followed by the PK studies in non-human primates. And when you do these studies, you can also do the PK companion toxicity studies by measuring the cytokine release and monitoring the weight to have an earlier indicator of the safety profile. These works are all considered to be done during the discovery stage. All right, next I'm going to share a case studies regarding the discovery of the CD3 VHH antibodies and also the evaluation of these VHH antibodies by the construction of the T-cell engager. All right, so this is a screening funnel of the CD3 VHH antibodies. We start by immunization of a camelid with the CD3 extracellular recombinant protein dimer, the epsilon and the delta domains, followed by the library panning and the screening against the recombinant protein as a Jekyll cell. Initially we identified more than 20 hits and selected 20 of them confirmed by sequencing and the antibody purification and the confirmation of binding affinities. Then we select the top 5 leads through the in vitro functional assays, and we discovered that one of these leads showed pretty good cyno-monkey cross-reactivities. So that's why we decided to choose these only leads with the cyno-monkey cross-reactivities to the next step, the humanization, affinity maturation, and the confirmation of the in vitro assays and in vivo validations. So starting from this page, we're going to share some more data regarding this CD3 antibodies and the efficacy data when we put this CD3 along with the DAA target. Let's start with the data of the CD3 monoclonal antibodies. Like I said in the previous page, we did affinity maturation. So this data basically shows after the affinity maturation, the parental antibodies showed improved the binding affinity and improve the efficacies. So on the top left panel, this is ELISA flow cytometry binding assays. The light blue one, the AS39678, is the parental CD3 VHH antibodies. As you can see from the from the figure, after affinity maturations, the flow cytometry binding curves are significantly improved. And at the lower left panels, this is a biocore kinetics data comparing the parental antibody affinity versus the affinity matured variants. In average, this affinity is improved from 4 to 25 folds. And on the right top panel, this is a T-cell activation reporting assays when we coupled the CD3 VHH with a GPRC5D VHH to form a typical T-cell engager with a tetravalent format. Again, this light blue one is the parental, the AS39678. After affinity maturations, the T-cell activation level significantly improved comparing to the light blue, the parental ones. In addition to the binding affinity to human targets, we also evaluate the binding affinity to cyno-monkeys. So this data shows by ELISA on the left and the flow cytometry on the right that the affinity matured candidates showed significantly improved binding to CD3 proteins and the cyno-monkey PEN T cells. So after the affinity evaluations, next we'll move on to the epitope information. On the left panel, we did a epitope competition by flow cytometries to compare the epitope of our candidates versus several other clinical benchmarks, including SP34, OKT3, and UCTH1. Judging by the competition profiling, you can conclude that our candidates shared similar epitope as the other clinical benchmark against the CD3 antibodies. All right? And in addition to this binding profile, we move on to the second part, the developability of these assets, these VHH antibodies. So on the right panel, we did a thermal stability test with the BsAb, and judging by the TM temperatures, our assets, the VHH CD3 antibodies shows above 30-degree TM1 values, which is significantly higher than the other clinical VHH benchmark. In addition to the stabilities, we also assessed the non-specific binding by DNA and BAP ELISA, and also the self-interaction by the AC scenes. I will not go through the data, but the conclusion is in these assays, these CD3 variants after the affinity maturations shows reduced the non-specific binding and reduced the self-interactions. In addition to that, we also used an AI software to predict the immunogenicity of these VHH CD3 variants. In these simulations, we included several clinical benchmarks as a reference for high risk and low risk of immunogenicities. And judging by the predictions, we can see that our assets to the left showed a medium range of immunogenicity risk. All right, so far we have seen the profiling data for the CD3 VHH monoclonal antibodies. And the next step, we're moving on to the bispecific by fuse this CD3 VHH to the CD20 TAA monoclonal antibodies. So as I mentioned previously, the majority of the T-cell engager utilize the monovalent versions. But here, in this case study, we actually tried several dozens of different formats, including monovalent, bivalent, and tetravalent. And what we discovered is with the proper selection of the VHH CD3 antibodies, tetravalent bispecific antibody is not always a nightmare for the safety issues. So the data I'm showing in the next couple slides all comes from these tetravalent formats listed on the right panel of this page. Basically, we have 2 copies of the CD3 VHH antibodies and 2 copies of the TAA arms in this symmetrical bispecific antibodies. The red circle represents the CD3, and that dark green represents the TAA arms. So the first data is the RGA assays. We did the RGA assays with 2 different cell lines. The left panel is with the CD20 positive cell lines, and the right panel is with the CD20 negative cell lines. On the left panel, we see robust activation of the T cells by the RGA assays. Some of them showed similar activation level to the clinical benchmark. And when we switch the target cell line with the CD20 negative cell lines, there is a minimal activation of the T cell, showing the proper safety profile of these candidates. Next, we'll move on from the RGA assays to TTCC assays with pan-T cells as the effector cells. Again, on the left column, the top is the TTCC assays with TAA positive target cell line, and the bottom left is the TTCC assays with the TAA negative cell lines. On the top, we see robust activation of the T cells comparing to the clinical benchmark, the glofitamab, while in absence of TAA expressions, there are minimal activation of the T cells, again showing the pretty decent safety profile of these candidates. And in addition to the TTCC, we also monitor the cytokine release, the induction levels in these assays, including the TNF-alpha, the IL-2, and the interferon gammas. Again, in these 2 different conditions, our assay showed much lower cytokine induction levels comparing to the glofitamab in the presence of the TAA positive cells and in the absence of TAA positive cells. So after the completion of the cell-based assays, we'll move on to the animal models. This is a pretty standard human PBMC transplanted CDX models. In this experiments, we compared 4 of the formats, the F28, 29, 30, and the 32, along with 2 clinical benchmark. And again, in this experiment, we see some significant inhibition of the tumor growth. Some of them show similar, even better than the clinical benchmark in terms of the tumor size. In addition to the efficacies, we also monitor the PK profiling of these candidates by doing this PK analysis in the rodents. So this is a typical in vivo stability test of the candidates. And judging by the half-life data, we can see that the half-life of our bispecific antibodies share a similar PK profile as other VHH-based bispecific antibodies, demonstrating pretty good stabilities of these bispecific antibody candidates. All right, so that's all of the data. Let's just do a really quick summary of what we have seen so far with these case studies. First of all, the humanization of administration was successful with this CD3 VHH. The affinity is significantly improved. totally 26 affinity matured variants were categorized, and the 6 of them were selected as the top candidates, showing a spectrum of binding affinity and the potency in the T cell activations. And again, the CD20 and the CD3 VHH bispecific antibody with different kinds of formats were constructed. And even with the bivalent CD3 arms, these antibodies showed comparable tumor cell killings with lower cytokine release in the TDCCSs and the potent tumor growth inhibition in the CDX models. So that brings to the conclusion that the bivalent CD3 and the TAA arms forms a tetravalent format, significantly improve the developability and the manufacturabilities comparing to the other TCE formats without any compromise of the safety profile. All right, to conclude, just a really quick introduction regarding our bispecific antibody discovery platforms. Currently, ProBio provides a one-stop solution of bispecific antibody discoveries, starting from the lead generation of monoclonal antibodies all the way to the preclinical candidates, including designing, construction of bispecific antibodies, the efficacy studies, and the pharmacology studies, and the developability studies. So far, we have delivered more than 40 bispecific antibody projects, with 2 of them in clinical trials, and hopefully our experience and expertise can help you to accelerate your pipelines with bispecific antibodies. And thank you all.
Thank you, and we will take questions at the end of the session for all the speakers. So I'll quickly move on to introduce the next speaker. We have Dr. Raymond Lee from BsAb. Dr. Lee is a distinguished researcher who has revolutionized our understanding of alternative splicing in cancer. His innovative methodologies have not only earned prestigious grants but have also opened new pathways in cancer research through his work at both Duke NUS and IMCD.
Thank you. Thanks the organizers for organizing this talk, and thank you for your attention. Okay, I'll tell you a little bit about our company and sort of like what we do. Okay. So we at PEX, right? We are a company that looks at alternative splicing, and the reason for this is that we believe that there is this critical need for cancer selective targets. So the critical question that we are trying to address, right, is this scarcity of novel cancer selective targets for the development of selective biologics that basically is a limiting factor in effective treatment options, right? And what we do is that basically our platform, right, allows us to identify cancer selective and highly prevalent tumor selective variants to basically develop therapeutics around, right? And what I'll show you in the next couple of slides, right, is that basically why we believe that splice variants are good targets, and it's basically a missed opportunity in therapeutic development. Okay.
Sorry to interrupt. Maybe you can move closely.
Okay.
Yes.
Hello. Can you hear me? Yeah. Okay. So this slide basically shows you the overview of what alternative splicing is, right? And basically, over 90% of the human genes actually undergo alternative splicing, right? And this process, right, gives you the opportunity for the genome, right, to actually encode a greater repertoire of like proteins, right? And in disease, what happens is that splicing can lead to pathogenic isoforms of genes. Okay. The pointer doesn't work. So on the left panel, right, what you see is that basically you have one gene, and after transcription, what happens is that you basically get a pre-mRNA. Right? And what splicing does is that basically you have this machine called spliceosome within the cell, and what it does is that it basically allows a single pre-mRNA to basically give you different transcripts, right? And how it does this is that basically it regulates the pattern of splicing, right? It basically selects junctions to basically join, and basically through this process, right, it actually controls inclusion or exclusion of particular sequences. right? And what this does is that basically this process allows you to have one gene encoding multiple variants present in the cell, right? And what you see on the right panel, right, is basically the multiple transcripts that are present in the cell that is derived from a single gene, right? And in disease processes, right, what happens is that basically the process of RNA splicing can be dysregulated, and you basically can have things which are transcripts which are cancer selective, right? Okay. So what we do at PEGS, right, is that basically we find that splicing is deregulated in cancer, and this is basically a known thing in the literature, right? And there's been basically many reports of the splicing changes in different cancer types. But however, the one of the bottlenecks for utilizing splicing for therapeutic development, right, is that there is actually no computational pipeline that allows you to basically identify the protein sequences that comes from these splicing changes, right? So what we have done at PEGS Bio is that we have actually developed a platform that allows you to look for this so-called cancer selective sequences and use that for therapeutic development, okay? And basically this next slide, right, shows you how we actually achieve it, right? So sort of on the cartoon on the right, what you can see is that basically there is a gene, and again, you can kind of see that there's like multiple transcripts being transcribed originating from a single gene, right? And what you see is that basically the exons which are colored in orange are the exons which are cancer selective, right? And what we do at PEGS Bio, right, is that basically we try to identify the exons which are disease relevant, right? And by doing that, what you can do is that you basically can get like a little piece of sequence. You basically can determine the consequence of splicing. And what you can find is basically you can identify a sequence that is cancer selective and use that sequence for therapeutic development, right? So this is what we do at PEGS Bio, okay? And in this next slide, right, what I'm trying to like impress upon you is basically sort of like the proof of value that that you can actually drug this so-called cancer selective variants, right? And what I've shown you here is basically two clinically validated cancer selective variants. One is called FGFR2b, which is a TKI, and the other is basically claudin 18.2, right? And what is interesting about these two variants, right, is that basically there is other variants that exist in the cell that is not cancer selective, but these are the variants that people are targeting in the clinic, right? So for example, FGFR2b is utilized, is basically targeted by this product from Amgen, bemarituzumab, right? And basically it targets the alternative splice region, right? Sort of like the orange loop that you see in the sequence. And the same thing happens for basically claudin 18.2, right? Where basically Astellas has an antibody that targets the alternatively spliced region, right? Which is shown in the orange piece of sequence at in claudin 18.2, right? So at PEGS Bio, right, we currently are developing a pipeline of antibody-based therapeutics targeting novel and cancer-selective variants, okay? So this next slide, right, gives you an overview of the things that we value in the targets that we identified, right? So as I mentioned earlier before, right, the platform basically looks for cancer-selective variants, and it also looks again for things, variants which are prevalent in the disease indication, right? So I will talk more about this point in the subsequent slide. But another key point of the platform, right, is that basically it identifies sequences that can be targeted by a biologic, okay? So this next slide, right, shows you basically the commercial opportunity for various targets in colorectal cancer, right? And what we think is particularly for cancer selective variants, right, it actually offers you the opportunity for therapeutic impact in larger populations than those with specific oncogenic mutations, right? So what you see in this chart, right, is basically two pie charts showing you the actual mutations in colorectal cancer, right? On the right pie chart, what you see is that other than KRAS mutations and sort of like BRAF mutations, which are basically present in like about 10% of those patients, the other actionable DNA mutations in colorectal cancers, right, are present only in single-digit % of those patients, right? Whereas the targets that we find for which are cancer-selective variants in colorectal cancer what we find is that some of these variants can be present in about 30% of those patients, right? So this represents an opportunity to actually make a bigger therapeutic effect in the cancer patients, right? Okay. So in the next two slides, right, what I want to show you is basically this proof of concept, right? And basically what we did to basically show that splice variants are clinically relevant, right? So this was basically the study that we initiated when I was in Duke NUS, right? And sort of like how we sort of like spun out the company, right? So we started off by trying to address this question of how or whether cancer patients actually mount a T-cell response to cancer-selective variants that are present in the tumor, right? And the answer to this question is yes, they do, right? And I will show you what I mean by that, right? So during the study basically started by looking for cancer-selective variants, which is sort of like what you see in the middle of the chart, right? The cancer-associated splice variant. So we found in head and neck cancer patients that there was this particular splice variant that was present in about 26% of those head and neck cancer patients, right? And what we can find is that basically this variant, right, is kind of like only present in tumor samples and not normal samples, right? And basically we asked this question whether cancer patients that have this variant, whether they were mounting an immune response towards that particular splice variant, right? And how we did it is that basically we took the sequence, right, from this cancer-associated splice variant. We look for peptides that can be presented by HLA molecules, right? And what we found is basically there was this particular peptide that we found in this splice variant. And then the next thing that we did was that basically we synthesized tetramers, right, which basically presents this variant peptide that we found, and basically look for T-cell responses in patients, right, in head and neck patients, whether they could actually detect T-cells that recognize that particular antigen, right? And what we found is that basically head and neck cancer patients have T-cells that recognize this particular peptide HLA complex, okay? Then, so this is sort of like this is the final slide that I'm going to show you. And basically it's a summary. It's a summary of the data that we found for this particular study, right? So previously, what I've shown you in the previous slide was that basically we could identify patient T-cell responses to the target. And how we found it was that basically we could do this tetramer staining for T-cells.
Yeah, it's fine.
Huh?
I think everyone can hear you.
Okay, sorry. Yeah, so what I was saying is that basically in the first part of the study, right, what I've told you is that basically we found a cancer-selective variant, right? And what we've basically demonstrated in the first study was that cancer patients actually have T-cells that recognize this particular cancer-selective variant, right? And then subsequently we built upon that observation to further validate the relevance of the observation by looking for whether cancer cell lines, right, that basically have that splice variant, whether they could sort of like present this peptide that we predicted from the variant, right, on the cell surface, right? And the answer to that question again is that we could confirm it using a mass spec experiment, right? That basically the peptide that we identified could be present on target cells, right? Then subsequently, the next piece of like data that we collected was that basically we took T cells, right, again from cancer patients. We basically expanded those antigen-specific T cells, and we asked the question whether the T cells could respond to those peptide MHC complex, right? And the answer again, what we saw is that when we did this stimulation experiment was that basically we could see T cell expansion of those antigen-specific T cells. So again, what this tells you, right, is that basically those T cells that are present in those patients, right, are somewhat functional, right? And finally, the final piece of data that we collected for this particular study, right, was that basically we generated T cells receptors against this particular peptide MHC. And what we find is that basically in this setting, right, when you take this TCR and engineer it into a T cell, we could actually demonstrate cell killing of target cells that basically express the splice variant, right? And in this case, what this basically shows you, right, is that basically splice variants are clinically relevant, right, and basically can be utilized for like therapeutic development, right? So basically, this is kind of like what are the things that we do at PAX Bio, right? So yeah, that's kind of like the end of my talk. Yeah, thank you. Thank you.
Raymond. I'm pleased to introduce our next speaker. Wait for the slide to come up. So Tina, whose regulatory experience has been instrumental in several breakthrough approvals. Most notably, she played a crucial role in expediting China's first COVID-19 antibody approval, achieving what typically takes several years in just 2 years. Her leadership has been transformative, particularly in building robust regulatory teams from the ground up. Tina, please.
Hello, thank you everyone. I'm so happy to be here to share with you some regulatory consideration during your CMC work, especially for the BLA stage. Before my presentation, I would like to share with you some track record of ProBio. ProBio has 2 business lines. One is biologics and another one is CGT. So in the past few years, we got some sound records. And for biologics, we have got more than 50 IND approvals. And for CGT, we got more than 70 IND approvals. And now for biologics, we will file a BLA very soon in October. And for CGT, actually, we filed 1 BLA last year. And we completed the on-site inspection early this year. So I'm waiting for the great news very soon. Yeah, today's my topic is focused on CMC consideration. The first part, I would like to share with you some general CMC consideration for BLA, also apply to bispecific and ADC product. And the second part, I would like to share with you some special considerations for both 2 kinds of products. First one, yeah, because I'm a RA person, so I would like to use IND or BLA package to talk about what's the regulatory agency's principles when they review the IND or BLA package. And this is a standard triangle for about the CTD. I think most countries use CTD package for their IND and BLA. So CTD contains 5 modules. For CMC part, the main part is module 3, the quality. And for the module 2, one aspect is the summary of the quality. For the main body of the data, module 3, they are on drug substance section, drug product section, and also appendix. And because we talk about the CMC, chemistry, manufacturing, and control, so of course the actions DS and the DP, we have manufacturing, and we also have control. And except these 2 parts, we also, because we use a lot of materials, so we have special sections for starting materials. And also for product, no matter which stage it is, the safety is very important. So we have a special section about the safety. Okay, so let me, in the next few slides, I would like to talk about on the manufacturing and control. The first, so as mentioned that for CMC part, we have materials, manufacturing, and testing. So for the materials, especially for some biologics source, we have special requirements. And for the manufacturing part, we have to show the agency how you design the manufacturing and how you do the control. And for the testing, not only the drug substance and the drug product, we also have some intermediates. So we also do a lot of testing on intermediates. Another part is about the safety evaluation. Safety is not just the testing. We should have a comprehensive control strategy from very beginning, starting from the materials. And of course, we have some safety control strategy during the manufacturing and also testing. So for the materials part, I cited the requirements from the ICH guideline for the CTD. Here you can see for the materials, we have to list all materials used during the manufacturing to agency, especially for some biologic source product materials. Like host cell, the history should be clear stated in the section, and also we do some testing and characterization for host cell. Especially vector, how you design and the sequence. What's the sequence? Is it the sequence you required? And is there any mutation? And for the cell line, because you will have production, so cell line should be chosen. The developing history should be also listed. And because we do the monoclonal antibody and biosafety others, so the chronology is very important. In all the BL package, we should clearly show the chronology data. And the cell line should be stable, so there are many passages. So what's the stability about cell line? Cell line is also very important. After the cell line, when you get the final clone, we have to create the cell banks. For cell banks, also, we need to do a lot of testing to demonstrate that the cell line is safety, no virus contamination. So, the testing according to different regions requirement when you submit the IND or BLA. And also because the cell line needs to be stored for a long time, so storage stability is needed. At the BLA stage, a lyrical study needs to be performed to define the limited cell age. Except these other materials also need to be show what kind of the spec and which manufacturing purchased from, especially for the animal and human origin. If there is animal or human sourced, TSE, BsAb variation should be assessed. So we have such kind of table to list all the materials in the filing package to show what kind of the specification and whether there is any TSE, BsAb risk and so on. For the manufacturing process, here is the life cycle manufacturing process validation. They are according to the agency's guidelines, 3 stages of the manufacturing process. The first one is process design, and the second one is process qualification, and the last one is continuous process verification. For stage 1, it's process design. It including the process development and process characterization. So when the project is in the IND stage, you may just use your platform process. There's no much optimization because you want to initiate your clinical trials very soon. So but when the project goes to the clinical stage, you need to evaluate whether the platform process is okay for the commercial. So because at IND stage, you want the timeline is the most important. So that time you didn't do a lot of process development work. So after in the clinical stage, you may need to optimization and then characterization will be performed before the PPQ run. So after process characterization, you will have a defined CPP, QPP like such process parameters and as well as control strategy. After that, the process qualification, the PPQ will performed during this stage. PPQ is not just a 3 consecutive production, it's a comprehensive work including like the facilities qualification, method validation, and other supporting validations. I have another slide to show the supporting validation. And when you got the PPQ data, a BLA file will do. The BLA filing is not the end of the process development. Even after your commercial, during the commercial stage, because the technology will always change, right? and also you will know more about your product and process. So there might be some change or process optimization. So continued process verification will be happened during this stage. And this is the polling validation, except the 3 consecutive product. I think not 3, at least 3 consecutive production. Other studies will be happen at the same time, such for DS, there are barrel class study, impurity clearance study, holding time, and also others. Like the resin and the membrane, it's not a single use, it will be multiple use, so the lifetime and the carryover study will be performed as well. And also, during the DS and the DP productions, there are a lot of consumers and also container corrosion system. So ENA risk assessment and the study will also performed. Of course, for DS and DP, there are shipment, so shipping validation will also happen. So after the materials manufacturing, there's control, quality control. For all the materials will be used for human use. The CGMP compliance is very important. All materials need to be produced under the CGMP condition. For the quality part, there are characterization, release, stability. Of course, there are analytical methods. So for the characterization, we need to study the primary structure, secondary, and also high structure. So except this potency, purity also need to be studied. For the release spec, when you're in the R&D stage, the specification may be wider, just use your platform specification. But when the product goes to the BLA stage, because you have more data, you know your product more. So the specification might be adjusted to have a stricter criteria. So for the method, when in the R&D stage, the qualification is enough. But when the product go to the BLA, before PPQ, the full method of validation is needed. And for the container corrosion system, as mentioned, the leachable and the extrudable studies to be performed. For many agencies, the leachable, extrudable studies for the DP container corrosion system is needed. But some countries is different, like in China, the DS container corrosion system also needed to perform the leachable and the extrudable studies. Different countries has different special requirements. And for the stability panel, as the ICH guideline mentioned, there are long-term stress and auxiliary studies. Except that we also have in use the stability, shipping validation, shipping stability data. These are all the general regulatory consideration for biologics. It's also applied to biospecific and biospecific antibody and also ADC product. So except these general requirements, I also would like to share some special consideration. This slide, I don't want to talk more. You are expert. There are many kinds of, there are different kinds of biospecific antibodies like IgG-like or non-IgG-like. So for the biospecific antibody, the requirements also need to do all the CMC work as I mentioned in the first section. And another part is the quality part is the agency's focus on. FDA issued one regulation about the biospecific antibody. In this section, it mentioned that quality attributes that may affect pharmacology should be studied, including the agent-specific affinity and like on and off rates and so on. So for this guidance, FDA said because compared to like monoclonal antibody, biospecific antibody, the stability is not so stable as monoclonal antibody. So for bispecific antibody, we need to pay attention on the stability, like whether there's a fragmentation, aggregation, and whether this will impact the immunogenicity, as the doctor mentioned just now. And also we need to have more study on the antigen specificity, affinity, and these items. So here I would like to stop to say that for the bioassay method is one key items need to be special pay attention. So for the bioassay development, this is the basic requirements. When we to develop a bioassay, we need to think about 3 items. One is whether it's MOA reflective, and another one is stability indicating. And also, as a release stability, we need to make sure that as a release method, we need to make sure that the methods, process, and accuracy is well controlled, so that the different batches can use the same method. So, ProBio has the integrity toolbox for these solutions. We depend on based on the drug MOA, like whether it's binding or neutralization, and whether the immune stimulation. So we choose different bioassay method, and also because develop different bioassay method, it needs different time. So we will choose like binding method for IND stage, and we will choose cell-based assay for BOA stage. As mentioned, that there are different kinds of biospecific category. So we have like for binding activity, we have 2 single target binding assay or dual target binding, and also same as the function potency. Whether to choose binding or function depends on timeline and cost effective. But generally, for different agencies, for when that in the IND stage, binding can be chosen, but function potency need to be developed for the BOA stage. There are several cases which from FDA. This is the first one. The project is in human study. The sponsor asked whether the binding method, dual antigen ELISA binding assay is okay for the sufficient for the initial phase 1, and the FDA replied that yes, it's acceptable. But they also mentioned cell-based assay will need to be developed prior to initial pivotal studies, and also suggest sample retain is needed to enable bridging study. And the second one is the end. The project is at the end of phase 1. It's also asked whether the 2 independent ELISA binding assay is okay for the phase 2. Generally, FDA agreed, and also the same they recommend a cell-based potency assay need to be directed for the pivotal studies. And the third one also it's end of phase 1 and the end of phase 2 like this. It's about the comparability. Similarly, the cell-based potency assay reflecting the in vivo mechanism of action should be set by before the pivotal studies. After the biospecific, this one is about the ADC product. ADC, as the name, there's antibody drug conjugated together. So they are small molecule and also biologics. So they are on several paths. Naked antibodies, linker payload as the intermediates, and also when after the conjugation, there are drug substance and the drug product. So in my very beginning the slides, it shows CTD section. There's one section about the DS and one section about the DP. But here, because we have antibodies, linker payload, and also DS, so how to draft the document is a question. I listed here, there's two options. One option A and option B. For option B, we have separated 3 DS section, one for monoclonal antibody, and the second one for the linker payload, and the last one is for the DS. But also we can choose option A. The antibody and the linker payload can be as intermediates to be included in the DS section, the S.2.4. But the content should be the same. You still need to mention everything about the monoclonal antibody and the linker payload. And for the drug substance and the drug product, they have the special quality items needed, like free drugs as the impurities, unconjugated antibodies, and the lactar rate. This is the quality control strategy for the ADC product. As mentioned, because we have the antibody, we have the linker payload, and also DS and DP. So for each items, you have they have their own quality control strategy. They have own the acceptance criteria. All items need to be released as stated the acceptance criteria. And also, because the change is very often during the product development, even in the clinical stage or after commercial. So when there's change, we need to do the compatibility studies. The comparability studies will be designed based on the change category. If the change just happened in the monoclonal antibody, we need to evaluate whether the change will affect the drug substance or drug product. If no effect, maybe comparability on the MAP intermediate level is sufficient. But if the change will impact the DS and the DP, so from the antibodies section and also DS and the DP, everything should be performed as a comparability studies. And for the comparability, we need to evaluate the changes, just the process or quality, or just the release data is okay, or also need the stability data, case by case. Yeah. So for the ADC product, because there are so many items, so it will be trigger the complex studies. Yeah, this is just a case. Maybe you can just for your reference how to perform the compatibility studies for ADC. Yeah, that's all my things. Because sorry, it's very quickly because of the time. Thank you.
Thank you, Tina. Our final speaker brings a unique blend of scientific expertise and strategic insight to antibody drug conjugates. His journey from medicinal chemistry at prestigious institutions like Oxford and Novartis to commercial strategy in APAC, and now leading cutting-edge ADC development, gives him a comprehensive perspective that bridges scientific innovation with practical implementation. Ben from Hummingbird Bioscience.
Cool, thank you. Thank you for inviting me here today. I mean, this microphone makes me a little bit nervous. Also, the fact we're at the end of the day, I want to make sure we have lots of time for questions. So I'll probably go a little quickly, but maybe we can dig in deeper during questions or afterwards. Feel free to grab me and we can talk a little bit more. So I think people know Hummingbird well enough. I think what I wanted to focus here today is on the kind of building out of our ADC platform and pipeline for oncology, and particularly what kind of led up to the deal with Cadeo Therapeutics, our sister company, and spin out earlier this year. So I was engineering this talk towards the complex modality space. So we work in kind of a multi-payload ADC space now. It's, as introduced by a previous speaker, very complex. I think we're adding another linker payload in. So I wanted to make sure that we were kind of well grounded in the fact that having a modality, having a solution is one thing, but really having a concrete understanding of what the problem statement is that we're trying to solve, that rationale, the opportunity basis for having a complex modality is super important. But more than that, once you've got to a point of inflection that we have of kind of that capital raise, the investment, the go ahead towards clinical development, really being able to execute that complex system very, very quickly and get to your clinical readout. Because as we all know, the in vitro data doesn't really correlate well to in vivo doesn't really correlate well to the clinic. What you really want to see is that clinical proof of concept and that value proposition brought out towards patients. So for us, it was looking at the landscape of ADCs a few years ago, seeing the kind of widespread endorsement of topoisomerase-based ADCs and others, and seeing really what was a large proportion of patients progressing on those therapies, with mechanism of action, particularly the payload being a key cause of resistance, and that there were no later line therapies that existed. We saw the opportunity to look at how to address that. And for us, that solution was looking at resistance and trying to resensitize patients or sensitize patients who were not initially treated with this kind of ADC back to the original mode of action. And then thinking of how to do that. There are many ways you can administer a couple of agents, but for us, doing it in one shot, a single agent approach with two payloads attached to an ADC seemed to be the optimal one. And I think our data and kind of the spinning out of our sister company has kind of proven that. Now, as we've done that, our focus is on, okay, now we have this, how do we get through the barriers and hurdles that might be there to quickly develop these ADCs, get to the clinic, and prove that our hypothesis kind of bears fruit for patients. So as I said, we looked at the landscape of ADCs, as everyone has. Widespread expansion in the last few years has kind of fueled a craze for preclinical assets, looking at a variety of spectrums. There are lots of variables you can impact in ADCs, typically looking at different targets, looking at different payload modes of actions, but this has spawned a huge number of preclinical ADCs. Despite this, there are two key challenges that remain. The first is the breadth of patients that are already addressed in indications with ADCs is quite small. And even for those that are addressed, patients progress very, very quickly with no later line of therapy that really helps them. Being a victim of their own success, you can see that the response rates are very compelling, but in the majority of indications on HER2, patients will progress within an 18-month time frame. And there are very limited therapeutic options afterwards. We moved quickly to look at kind of the key clinical data that was emerging a few years ago, and it was clear that HER2, the target for HER2, was still expressed and retained by the majority of patients after they become resistant. This pointed us towards the fact that it's a mechanistic resistance towards the payloads that are used by topoisomerase-based ADCs. This pointed us towards the fact that HER2 in this situation remains quite a valid target, and this has been shown before in sequencing patients between ADCs. So TDM1 was developed before TXD, and clinical work showed you could treat with another ADC targeting the same antigen on the cell surface and see good response rates. Taking this forward, we thought about how can we best treat these patients. We could try to switch agent to a different mode of action. In theory, works very well. In practice, there are very few proven, validated modalities out there. It's only a handful of mechanisms, a very limited scope, and also these are typically shared by earlier lines of chemotherapy, so you're likely to still encounter resistance when you try to switch to a different modality. For us, it was really moving towards a combination. There's plenty of early-stage clinical work that has shown hints of efficacy, sometimes limited by combining chemotherapy and seeing toxicity. Can we combine two agents to either target orthogonal modes of action, or can we combine two agents where one would kind of resensitize patients back to the original cytotoxic mode of action, kind of akin to synthetic lethality? And this is where we chose to spend our time, as we felt this had the most potential traction. We looked at public domain gene screening, where we could identify potential targets, gene targets, which would show the greatest ability to sensitize tumor cells back to the cytotoxic mode of action. So here on the left is basically a screen where you knock out various genes and see the sensitivity to that with the original payload. On the right, we took these gene targets forward. We looked at kind of ideal looking payloads, did some in vitro synergy screen work to show that one, this public data set did bear fruit in our hands when it came to payload screening. But also they were synergistic within a good range and a good kind of molar equivalency. As we'll see later, that's kind of an important design consideration if you're bringing two agents on one single therapy to a cell. There are many ways you can kind of tackle this. Folks have looked at ADCs plus chemo. Folks have looked at ADC plus ADC. And we were looking, as were others at the time, at kind of dual payload ADCs, which seem to be the most attractive from a range of different criteria we may consider for a therapeutic. In terms of tolerability, administering one agent is much more preferable, especially over a systemic therapy. Having complete control in terms of temporal and spatial delivery of the payloads to a tumor cell also preferential. Ease of clinical development, although complex in nature, it's still a single agent when it comes to clinical trial, which can prove effective and efficient for various reasons. Lastly, kind of the optionality to then include future agents as well. We have one agent that can potentially combine another. To do so, we are obviously leveraging our antibody platform that we've spent the last 10 years building up, but looked at also conjugation spaces and building our expertise there. We built a proprietary linker platform technology as when we surveyed the landscape. We felt that we could make a linker that was much more stable, much more homogeneous, and also flexible to kind of future payload combinations that we may envisage going into oncology and other diseases. And lastly, we had then, from the previous work, selected our kind of anchor payload combination for dealing with these post-Enhertu, beyond Enhertu settings. The picture on the left is from an older publication, but I think the key to some of our success has been in looking at the variables which impact ADCs the most. So one of those is conjugation site and linker profile. Changing where the linker is attached has a very dramatic effect on the half-life stability of your ADC. And we found as well, there's a particular privileged region where we can conjugate a linker well and see much enhanced ADC stability over other proven ADCs such as Enhertu, as we see on the right, where our hydrophilic linker system plus the conjugation site gives us kind of a dramatic increase in stability, which is needed for a more tolerable therapy, and to see high doses in the clinic. Back to the payload decision, we had a couple of classes in mind in payloads, but it's one thing that is key and it came out of our synergy work as well, is ensuring that the potency is quite well matched. Dual payloads are not intrinsically new on an ADC. They've been explored for over a decade, give or take, as long as ADCs have been around. But without A, a biological rationale to produce a complex molecule, and B, a rational payload combination, it's been very hard to progress. On the left was a very earlier piece of work to combine PPDs with another agent. As you can see, if you pay close attention to some of the green and blue lines, that you kind of get only PPD performance, both in efficacy, and the same would be expected in tox. PPD is orders of magnitude more potent than the MMA or a statin payload, and what you see is just the dominant payload driving anything that you see pharmacologically. Whereas on the right, a group recently showed that with an auristatin plus auristatin combination, they were able to achieve some level of synergy because of the relative potency balance between the two payloads. In fact, here they have kind of a non-cleavable and a cleavable one, some bystander effect, non-bystander effect. But they also showed that, as we have shown in our hands too, that the combination of single payload ADCs was inferior because of the kind of utilization of the same target. The last kind of slide on data I'll show, which led up to the licensing out of our platform to Calio Therapeutics at the beginning of this year, after kind of a solid year's worth of development of the ADCs, was the validation of our hypotheses, both in vitro and in vivo for our CDX models. So on the left, we screened our payload, a dual payload ADC, in a range of cell lines with different sensitivity levels to the topo 1 mode of action. And not only can we see kind of greater overall inhibition of cell growth, but that vast difference, especially on a low sensitivity cell line between a single topo 1 isomerase payload and our dual payload combination. This translated in vivo on the right, again looking at cell lines that are either intrinsically resistant or have acquired resistance to T-DXd, and cell lines with high levels of DNA damage repair activity. We can see that our dual payload ADCs outperform and are well validated in vivo. And this gave us enough confidence to go ahead and start preclinical development, looking at toxicity and beginning out licensing. So that was kind of a great moment for us, an inflection of value and proof of concept preclinically, but the next part is the how do you get to that clinical proof of concept quickly, and this is where we spend a lot of our time and focus now. ADCs, as been pointed out, are made of multiple components. We've added an extra one and potentially with another payload. This increases development, regulatory, and clinical complexity potentially, which can lead to significantly longer timelines than other simpler modalities. And with as well a good biological rationale data in the public domain, we see kind of a rapid expansion in other companies also exploring the payload space. How do you make sure that you're one of the first ones to reach clinical proof of concept? For us, we focus hard on development timelines, so preclinically making sure that manufacturing proceeds at risk and early. Kind of at risk investment, but something which then mitigates a very long pathway through development. We engage regulators as early as possible. There are many meeting forums now to engage various regulators well before your classical pre-IND meeting, where we can address items around manufacturing, around how do we deal with maybe two payloads or multi-payloads in an ADC setting, which adds a level of complexity. Also key has been developing an efficient clinical strategy where we can quickly get A, into the clinic, but also B, through early cohorts as fast as possible to get to that proof of concept point. And the last two are kind of key from a business point of view. How do we kind of minimize costs? For us, the landscape of vendors and acceptance of global clinical data is constantly evolving. And this for us allows us to minimize a lot of our costs and potentially put more than one proof of concept molecule through that early clinical stage and make sure that we have multiple maximum optionality in terms of how we develop our molecules. And the last point, given the landscape does change quickly and folks do catch up, maintaining an understanding of our differentiation. So back to that problem statement, where was the opportunity and how differentiated are we compared to the others that are emerging in the field or other modalities has been key to make sure that kind of the investment and cost to make complex modalities still makes sense. So that was a very fast whirlwind tour. I'm almost on time. I guess we're going to move to questions, but if I don't get to address any of them, feel free to grab me afterwards and I'll have the chat. Thank you.
I think we'll invite all our speakers to sit in the front, and then we can take the questions
一个
from
那个
the floor.
PPT 吗？就是这个，可以，谢谢。
Any burning questions? The audience? Yes.
Raise your hand, please. Don't be shy. Yes.
Thank you. I have a question for Ben. For the two payloads, so we understand you look at synergy, and it's kind of like straightforward, not straightforward. You look at how the drug works together after they penetrate in the first cell, right? But after, when you have bystander, when they are released in the tumor microenvironment, I mean, you have different stability of the drug, different decay. So how do you combine those aspects beyond the first internalization and the first release from the ADC?
Well, I think the first part is around design. So I think there is an aspect of what are you kind of trying to achieve. So I think other folks and us have looked at whether you want to kind of accumulate a certain payload or overcome a certain level of heterogeneity. Maybe there's different sensitivities across different tumor cells. I think there was one aspect around choosing payloads there. And the other aspect around the profile of the payloads, I think for us is looking at that initial synergy screen, is how they perform and whether it's necessary to deliver them and their mode of action simultaneously. I think that is the biggest question. The ultimate phase of those payloads, the different metabolism profiles, PK, I think that is more of an impact on safety than anything else. So is it very, very potent and want it to clear out? So in our circumstance, we have kind of the core kind of topo one payload, which we understand clinically and how that performs. And the other combination partner that we decided to go with has a lot cleaner profile. So we would look as well at things like overlapping toxicity concerns, do they hit different compartments, different tissues. In this case, it was much more of a kind of a clean bill of health. I think the challenge was making sure that those two payloads get into the same cell at the same time to drive the synergistic mode of action. So it comes back to design and what we're trying to achieve, basically, rather than thinking about what happens post-release.
Any questions?
Question for Derek.
Yes, go ahead.
The different formats of bispecifics that you have seen in your experience, between the 1+1 and the 2+2, I understand there's a difference, but within the 1+1 and the 2+2, any preference or any something that is more successful than others or something that is more failing than others in terms of formats?
Yes, so it's really up to what kind of CD3 antibodies you are fused in the T-cell engagers. We previously did a lot of trial with the SP34 variants by fusing the SP34 Fab with the TA antibodies. Under that circumstances, the 1+2 or the 1+1 is are the ones that gives the best balance between efficacies and the safeties. But however, in the data I just shared in the slide deck, when we switched the SP34 with the VHH version with anti-CD3 antibodies, because that one was a lot weaker. Well, not a lot, but a little bit weaker than SP34. And it's VHH instead of the Fab. So I assume there are a little bit difference in terms of the binding or the agonist activities when encounter with the CD3 proteins. So that's why when we include the bivalent of the CD3s in the bispecific antibodies, we're still able to see a pretty good balance between efficacy and the cytokine release. So to answer your questions, again, it's really up to the profile of the CD3 antibodies of your choice.
My question's for Tina. So in the ADC side of kind of the regulatory submission, you presented option A, option B. One option was very big dossier, the other one was kind of individual intermediate dossiers. I was wondering how much kind of development strategy plays into which one you would decide. If you wanted to use, say you're saving like a payload in the future, you wanted to maybe file for a drug master file, would you go down option B or would you go into option A? I wanted to know how much that plays into it.
Actually, as I mentioned, that no matter which option you will choose, the contact should be the same. Like you mentioned, for the linker payload, sometimes there's the DNF, so we can just no need to submit the documents anymore. We just ask for the LOA from the vendor, and that the agencies can revert to the DNF. But that is for the FDA filing, because FDA has DNF. But for some countries like EU, we don't have the DNF, so we still need to provide the whole section for the just map and also for the linker payload. But I think for the option A and option 2, you can see there are still 7 sections, so still need to provide the manufacturing quality. These items just put as a separate section or just included in the DS section. The content is the same, but for FDA, yes, DMF will be easier.
Okay, thanks. This is a question that several of you could jump in on. For ADCs, would it be more interesting to have additional antibody targets or additional payloads? Or is it the combination of the two that could be unique in some cases? So where is the future direction going? We'll start from one side and we'll do that.
I mean, I guess I have like a bias, right? Because we started off as like a target discovery company, right? So I will argue that the target is kind of like the space that you would want to innovate, because obviously a lot of like how to say, like T-cell engagers and some of the ADCs, right? They are really like focused on like particular like targets, right? So like for example, like HER2 is like basically kind of like in some ways kind of like done to death, right? And the reason why people do it is that basically if they have like a novel linker payload, then that will be like the target to go for, right? And obviously it will be limited by sort of like what has already been approved, right? So you basically have HER2 as sort of like the first line, and what happens next is that it'll be difficult to kind of like challenge that, right? Because they have to kind of like fall off like in HER2 and then start on your new drug, right? And then that obviously makes it challenging, right? Yeah. So I think like from my perspective, I would sort of say that target is kind of like a space that is not so competitive, I would say, in some ways. But of course, the challenge there is basically how do you like prove that that is the target, right? Because there you really have like a big space, right? Yeah. And how do you be convincing? Yeah.
Well, that's a good question. I think our philosophy comes from a slightly different angle. So it's kind of two things, right? I think it's have you, and this goes back to our kind of some of the founding philosophy behind it, but have you kind of adequately targeted that antigen in the first place? I think a lot of the earlier work making antibodies which have been used for ADCs may not be the best in terms of internalization, the best in terms of ADAs, the best in terms of its profile towards making an ADC, such as hydrophobicity. So I think the question I have is like, have the kind of existing targets been optimally drugged? I think assuming that an antibody that was discovered 10 years ago would be great is probably a bit of a fallacy. And the other one for us is, yeah, I think folks do kind of tend to hone in on the same targets again and again, but there's definitely a reason there. I think that's, again, back to one of my slides, is that patients still progress. I think we haven't found that magic payload or payload situation which deals with all indications. And I think we can see it, if you look at CRC compared to the breast cancer, we see that certain subsets of patients and indications just are intrinsically resistant in some way or challenging. So I think more targets can be beneficial, but I think, fundamentally dealing with the issues we have with the ones that we do have might be more beneficial. I think that's why we do so much work on the same targets again and again and again. I'd love to see more good targets, but for now the ones that we have just simply work very, very well.
For starter, I think I'm going to vote for the targeting issues, especially for the solid tumor. I mean, ADC has the similar issues, potential issues as the T-cell engagers for the solid tumor, as known as the on-target off-tumor effect, because most of the TAs we chose are universally expressed in both cancer tissues and also the healthy tissues. The only difference is the expression levels. So when it comes to ADCs or TCEs, it's a pretty challenging situations to find a way for the drugs to target to the cancer tissues with the TA expressions, but not the healthy tissues with the same TA expressions. So that's why I chose this one. Okay.
thank you all for coming. We have a couple of gifts for our invited speakers today on behalf of the organizers. Nassim, please.